guys, I'm going to show you a fast and safe and simple way to do DNA isolation from fungi. So what you need is basically you need your fungal pure isolates, which I have right here. You need the CTEP buffer, your 2-propanol, and your TE buffer. In addition to that, for this protocol, you would also need your plastic pestle for sort of grinding your mycelia that you get from your fungal isolates, and you'll also need your Eppendorf tubes where you put your tissue. In addition to that, you also need your inoculating loops, as well as your cut paraffin paper that you'd use to seal your plates after you have used them. So let's get right into it. So the first step that you need to make sure you get right is labeling your Eppendorf tubes. So I have them, as you can see, they're labeled. I have 2 isolates that I'm going to be working with. So I'm going to put my solution of CTEB in this first 2, and then my solution of true propanol in the second. So since I have 2 samples I'm going to be working with, firstly what I'll do is load these first 2 tubes with my CTEB buffer. So this is essentially the first step of the process. I'm going to put 0.5 mils of extraction buffer into the first set of 2 tubes, and then I'll go on to put 0.3 mils of 2-propanol into the second set of 2 tubes. That's in preparation for one of our final steps. So you can follow as you can see in the video. So this is essentially the first step of the process. So firstly, I'm going to load 0.5 mils of extraction buffer into the first set of 2 tubes, and then I'm going to continue and load the other set of 2 tubes with a 0.3 mils of 2-propanol, and that's in preparation for one of our last steps. You can follow as I continue to load my tubes in this video, and it's important that you put the right amount in the right tube so that you don't have flawed results at the end of the process. So various types of mycelia and conidia can be used as starting material, and in this case we're going to use fungal mycelia as our starting material. I'm going to collect approximately between 10 to 20 milligrams of fungal mass, and then I'll put this into my CTEB buffer, which is my extraction buffer. You can use an inoculating loop like the one I'm using in this video, or if you have a toothpick it could be easier. Make sure that you collect enough mass so that you get a good quantity of DNA at the end. In this experiment, agar fragments in the mycelia don't really affect the process, so don't mind scraping off agar pieces. They won't affect your results at the end. So you can follow through each and every step of the process as you can see in this video, and make sure that as you scrape off the mycelia, you do not contaminate your sample. So now we are done scraping off our mycelia. We've closed our plate and we're going to reserve it for future use. This is how our plate looks after scraping off the mycelia. So the next step is to try and mechanically pulverize our fungal tissue, and to do that I'm going to use a plastic pestle. So what I'm going to do is to gently push down my mycelial tissue and push it against the tube walls so that at least I try to break the cells. And fortunately for this protocol, complete pulverization is not required, so just try to do as much as you can and be gentle so that you don't spill your sample, and you are able to maintain a good quantity of sample material. You can follow as you can see in this video. It's a simple process, and just be gentle. This next step is fast and simple. We are going to eliminate cell debris and contaminants by centrifuging our cell lysates at 500 RPM for 10 minutes. This next step is simple and straightforward. I have taken my samples out of the centrifuge machine, and what I'm going to do next is to decant the supernatant directly into the 2 tubes of 2-propanol that I prepared in the first step, and then I'll discard the remaining lysets and cell debris after decanting. You can follow this step as you can see in this video. It's simple and straightforward, and make sure that you take as much supernatant as you can. Precipitate DNA, I'm going to centrifuge again at 12,000 RPM for 10 minutes. So now I have my samples after the second centrifuge. I have my pellet and a supernatant on top of it. And what I'm going to do next is try to discard as much of a supernatant as possible. Then after that, I'll go on and add 50 micromils of 1 times TE buffer, and then that's for dissolving the DNA pellet. And I'll have my dissolved pellet in the TE buffer at the end of the process. So now that I have my pellet at the bottom of a tube, what I'm going to go on and do is dissolve the pellet. So I'm going to add, like I said, 50 microliters of TE buffer into the tube so that I dissolve the pellet, and then that's pretty much the end of the process.